guys, there was some concern uh, around the fact that coronavirus can get mutated and the vaccine wouldn't work. And I just came across this article, and there's actually a preclinical trials going on this new nanoparticle vaccine candidate. So this is the nanoparticle vaccine, and it looks like this. Do I have a picture here? No, I do not. So let me show you the picture first before I get into further detail. So that's how the nanoparticle look like, and it is composed of two components, I-5350A and I-5350B. So the I-53 is um, fused to a RBD of this is the RBD receptor binding domain of the coronavirus. So the I-5350A component is fused to the RBD, and this is another component 12 times 20 times, and then that's how this is made. That's how this vaccine is made. I'm gonna go into more detail, page 5. But before I go into more detail, I'm gonna explain it to you how this virus is so different. So how this vaccine is so different. So this nanoparticle displays 60 copies of RBD that I just showed you in a highly immunogenic array and induce neutralizing antibody titer roughly tenfold higher than the prefusion stabilized S ectodomain tremor. So what is the pre-fusion stabilized S ectodomain tremor? So let me show you that. So coronavirus uh, have they have decorated coronavirus are decorated with a spike glycoprotein that binds to host cell receptors and mediates cell entry via fusion of the host and viral membranes. So there's a fusion that takes place between host cell receptors and the glycoprotein spike. So the idea of a vaccine is to create spikes, right? And they are pre-fused, obviously, because they haven't entered into a. They're pre-fused, and there's a S1 unit subunit that contains the RBD, and then there is S2 subunit, and this mediates membrane fusion and contains an additional protease cleavage site referred to as S2 that is adjacent to a hydrophobic fusion peptide. But you don't need to go into much detail. But so what they're saying is stabilized prefusion conformation conformation of class 1 fusion protein is desirable for vaccine development because this conformation is found on infectious virions and displays most or all of the neutralizing epitopes that can targeted by antibodies to prevent the entry process. So, so that's if you want to read more on it, how the prefusion class 1 spikes are made, you can do it. But there is a concern with that. There is a misfolding and that occurs that can cause, for example, a structure-based design of prefusion stabilized MERS-CoV and SARS-CoV spike ectodomains resulted in homogeneous preparations of prefusion spikes and greatly increased yields. These variants has to be contained two consecutive proline substitutions in the S2 subunit in a turn between the central helix. So this is more about this um, vaccine. It's a different candidate. It's not. This is a prefusion stabilized SARS spikes. These are just spikes. So if you want to know more about these spikes, you can go and read up on it. But the I'm focusing on this video on nanoparticles, and antibody elicited by the nanoparticle immunogens target multiple distinct epitopes on the RBD, suggesting that they may not be easily susceptible susceptible to escape mutation. So the antibodies that were produced by the nanoparticle immunogens target multiple distinct epitopes on the RBD, suggesting that they may not be easily susceptible to escape mutations and exhibit a significantly lower binding to neutralizing ratio than convalescent human sera, which may minimize the risk of vaccine associated enhanced respiratory. So, any vaccine associated risk is low with because of the low binding to neutralizing ratio. The high yield and stability of the protein components and assembled nanoparticles, especially compared to SARS-CoV-2 prefusion stabilized S trimer, suggests that manufacture of the nanoparticle vaccine will be highly scalable. So it is very stable and it has a high yield, uh, and and it is pretty stable. These results highlight utility of robust antigen display platforms for inducing potent neutralizing antibody responses and have launched cGMP manufacturing efforts to advance the lead RBD nanoparticle vaccine into the clinic. As the coronavirus as glycoprotein is surface exposed and initiates infection, it is the main target of neutralizing antibodies upon infection and the focus of vaccine design. As trimers are extensively decorated with aniline glycans that are important for proper folding and for modulating accessibility to host proteases and neutralizing apps. Vaccine development efforts responding to the COVID-19 pandemic have made extensive use of platform technologies for antigens design, antigen display, and vaccine delivery. Existing nucleic acid and vectored vaccine platforms enabled rapid entry into the clinic with vaccines encoding SARS-CoV-2 as antigens. However, the safety, efficacy, and scalability of these vaccine modalities are not fully understood as there are currently no DNA or mRNA vaccine licensed for human use, and the first viral vector vaccine was approved only within the last few months. In contrast, self-assembling or particulate protein immunogens are a clinically validated vaccine modality with a proven track record of safety and efficacy in humans. Recently, self-assembling protein platforms for heterologous antigen display have matured significantly, and several protein nanoparticle vaccine display viral glycoprotein antigens are currently being evaluated in clinical trials. In preclinical studies, vaccine candidates based on designed protein nanoparticles have significantly improved the potency or breadth of antibody responses against numerous antigens, including prefusion. Here we report designed protein nanoparticle vaccines multivalently displaying the SARS-CoV-2 RBD that elicit potent and protective responses in mice with neutralizing titers an order of magnitude higher at a 5-fold lower dose than soluble prefusion stabilized as, while also exhibiting a significantly lower binding neutralizing ratio than convalescent human sera. I-5350 is a computationally designed 28 nanometer 120 subunit complex with icosahedral symmetry constructed from trimeric and pentameric components, all amino acid sequences provided in supplemented item. The nanoparticle can be assembled in vitro by simply mixing the independently expressed and purified. So the important detail here is the RBD residues was genetically fused to I-5350A using linkers comprising 8, 12, or 16 glycine and serine residues. Nanoparticle revealed predominant peaks corresponding to the target icosahedral assemblies and smaller peaks comprising residual unassembled dynamic light scattering DLS and negative stain electron microscopy confirmed the homogeneity and monodispersity of the various nanoparticles, but both before and after freeze thaw, the average hydrodynamic diameter and percent polydispersity measured by DLS. Um, the average hydrodynamic diameter and percent compared to hydrogen deuterium exchange mass spectrometry confirmed that display of the RBD on the trimeric component preserved the conformation of the antigen and the structural order of several distinct antibody epitopes. Finally, we use glycoproteomics to show that all three components were N-glycosylated at positions N331 and uh, molecular surface representation of the SARS-CoV-2 in the prefusion conformation. Each prototomers is colored distinctively, and link glycans are rendered dark blue. Um, Antigenic characterization of SARS-CoV-2 RBD. We use recombinant human ACE2 ectodomain and 2S specific maps to characterize the antigenicity of the RBD when fused to one I5350A, as well as the accessibility of multiple RBD epitopes in the context of the assembled nanoparticle immunogens. We use biolayer interferometry to confirm the binding affinities of the monomeric humans. Uh, we previously observed that the magnitude and quality of nanoparticle immunogen elicited app responses can be modulated by the accessibility of specific epitopes in the context of a dense multivalent antigen array. No degradation, SDS page. Oh! Multivalent antigen presentation on self-assembling protein scaffolds is being increasingly explored in clinical vaccine development, largely due to the availability of robust platforms that enable the display of complex antigens, including oligomeric viral glycoproteins. These advantages include the potential to induce potent neutralizing ab responses, the ability to use existing worldwide capacity for manufacturing recombinant protein, and an establishment regulatory track record.